Hi, my name is Chiu, and today I'll be talking about liquid-liquid phase separation in space. So we know that space can have many different effects on the human body. However, one of the least understood impacts of space is the one on the brain. And so for this reason, I think it's really important to study these impacts in order to protect astronauts from developing potentially deadly brain diseases as a result of space travel. And one such disease is Alzheimer's disease, which is a disease associated with aging. As you can see, Alzheimer's disease is caused by neurodegeneration in the brain, and symptoms can include memory loss, behavioral changes, and difficulty in daily tasks. And disease onset is typically during the mid-60s or later. So as you can see, a healthy brain, it has more mass than a brain with Alzheimer's disease because of that neurodegeneration. And more specifically, a pathological hallmark of Alzheimer's disease and also other neurodegenerative diseases is the buildup of protein aggregates in and around neurons. For AD, tau aggregation is one of the major contributors to the progression of the disease because the aggregates can form around neurons and that kills the neurons. So liquid-liquid phase separation, going back to my title, is a very fundamental organizing principle of biology. It's a process in which a homogeneous solution of biomolecules separate into two distinct phases. And the concentrated phase separates out specific biomolecules and proteins together so they can, in a droplet-like condensate, so they can interact more freely with one another, and it allows them to catalyze biochemical reactions easier. And this process is very important to a cell's function. As you can see, there are many cell processes that use phase separation to function properly. And normally phase separation is a very liquid and dynamic process, and it's very reversible, but under some conditions it can become irreversible, like mutation or environmental stress. And this process makes phase separation irreversible, and it forms solid-like aggregates. And so you can see here tau as a liquid droplet formation, and then tau can aggregate into fibrils, which then surround the neuron and kill them. And furthermore, some mutations of tau can actually cause the rate of phase separation to increase. So the bottom two rows, you can see that the droplets are larger and they form earlier than the wild-type tau on the top row. And this is pretty dangerous since it can cause an earlier onset of Alzheimer's disease. So multiple articles have actually suggested that astronauts in space experience many age-linked symptoms like osteoporosis and muscle shrinkage or neurodegeneration. And so my hypothesis is that the environment in space increases the rate of phase separation, which then causes an increased risk of neurodegeneration. This leads me into my proposal for my Friesenfly, where I'll be comparing in vitro phase separation of tau on Earth and in space. To do this, I will synthesize tau proteins using the BioBit cell-free system from the Genes in Space toolkit, and in order to detect the amount of phase separation that occurs, I will use the N and C terminal fragments of Venus, which is the yellow fluorescence protein. And how this works is that when they're on their own, the fragments of the YFP can't fluoresce, but as tau interacts with one another during phase separation, they reconstitute to form a full-length protein which can fluoresce. And as you can see in this picture is tau under normal conditions, and this is tau with an environment that induces aggregation and phase separation. So you can see there's a clear difference between the two pictures. And in order to detect this, the amount of fluorescence there is, I will use the fluorescence viewer that Genes in Space will get. And so in order to compare the phase separation, I will have two main groups, wild type and mutant tau, in space and on Earth. And these two groups will help me compare and make conclusions about how the space environment affects phase separation. And even though my experiment uses YFP, you can see on this chart that YFP and GFP are actually pretty similar on the color spectrum. So it won't affect how the fluorescence viewer views the fluorescence. And this is a graph that supports my hypothesis is that phase separation is faster in space. So you can see the y-axis is the intensity of the fluorescence in each tube, which corresponds to the amount of aggregation and phase separation in each tube. And the darker colors are the test tubes on Earth, and I would expect to see those have less fluorescence and less phase separation than the ones in space. So I would expect to see the mutant space sample have the most amount of fluorescence and the wild-type Earth sample have the least amount of fluorescence. These are two other of the possible things that could occur from this experiment. If phase separation is the same in space, I would expect it to look like this. And if phase separation is slower in space, then I would expect the mutant Earth sample to have the most fluorescence and the wild-type space sample to have the least fluorescence. And for my ambitious experiment, instead of using synthesized proteins, I will use a mouse model with Alzheimer's disease and to observe the effects of space on the brain over an extended period of time. So I will have 5 separate groups of mice that will live on the International Space Station for different amounts of time, for 2 weeks, 3 weeks, 4 weeks, 6 weeks, and 8 weeks. And I chose these time points because the mouse model that I'll be using usually shows aggregation around the 4-week time point. So I included time points before and after 4 weeks to really monitor how space affects aggregates. And to read the amount of aggregation that occurs, I will use microscopy. So I'll take a slice of the brain and I'll stain it with antibodies that only recognize pathological tau. So what this means is that the antibodies will only bind to the tau that has aggregated, so that'll make it clear where the aggregated tau is. And as you can see in the wild-type mouse, there is no aggregation occurring. And this is one of my anticipated result for this experiment. The y-axis is the percentage of cells taken up by aggregates, and I expect to see that the mice living in space will have more aggregates in general than the ones on Earth. And if phase separation is the same in space, I would expect to see something like this, and if phase separation is slower in space, I would expect to see something like this. So in conclusion, if my hypothesis is proved right, it would have significant impacts on the future of space exploration because the increased phase separation would lead to more aggregates forming in the brain, which would mean that even people with wild-type tau would have a higher chance of developing neurodegeneration, and it would have an earlier onset, which puts more people at risk. And even if my hypothesis isn't right, I think that we can still learn something out of this experiment overall, because even though my experiment focuses more on the abnormal phase separation, I think that phase separation is still a very important biological process, and we can still learn many things about the human body through this experiment, even if my hypothesis isn't right. And I would like to thank my mentor, Dr. Michael Das, for helping me with this presentation, my sponsor, Mr. Hines, the Genes in Space team, and the sponsors. And yeah, thank you.